Hi, Andrea. How are you? I was hoping you could introduce yourself and tell us a little bit about your background.
Yeah, I'm Andrea. I'm a PhD student at the University of Pennsylvania. I'm currently working in the lab of Trevor Penning, and I am focusing on biochemistry and biophysics in my research. Our lab specifically focuses on toxicology within like that greater field.
Very good. I know you've studied the impact of environmental pollutants such as PFAS on hormonal regulation. Could you provide an overview of some of that work?
Yeah. So, like how I mentioned, we look at toxicology, and then you can further break down toxicology into different subfields of it. And one of them is environmental toxicology. So, environmental toxicology looks at contaminants in the environment to see how they impact human health or animal health, although we're interested in human health. So, we are studying the effects of PFASs. Our lab focuses on hormonal regulation as well. So we often hear about environmental contaminants that can impact hormones, and we talk about how this can affect our developing bodies. We often hear about it in the context of polluted milk or pollutants in the water and how they affect human hormones. And we call those contaminants endocrine disrupting chemicals, since the endocrine system is the system that deals with hormones. The study of endocrinology and all of hormone regulation is a wide one. To simplify it down, hormones are chemicals in our bodies that relay a signal to other cells that causes a change in the cell to produce whatever type of protein or whatever type of phenotype they need it to produce. So they're really messengers. And this happens because the chemical can interact with the receptor, and the receptor on the cell will then tell the cell what to do. However, there are ways to change the levels of hormones in our bodies. So, naturally, without any influence outside of the human body, we do have other chemicals that can regulate the concentrations of these hormones. That way, in a certain period of your life where you might need a lot of a certain type of hormone, you will be able to produce it. And then in other times of your life where you don't need it, it will be inactivated. And these are pre-receptor regulators. These enzymes can act on the hormone to produce it or inactivate it before it even reaches the receptor, which then elicits that biological response. So, our lab focuses on these pre-receptor regulators. And with all of that context, we come to the novel finding that we've had here. It's been long speculated that PFA, PFOA, which is a type of PFAS, perfluorooctanoic acid, is a type of per and polyfluoronated alkyl substance. It's been long speculated that PFOA can change hormonal levels or hormonal biological implications. It can have an effect essentially on androgen receptor. Androgens are a type of hormone. Androgens are often thought about as the male sex hormone. So you can think of things like testosterone, DHT, DHEA, which you might have heard in like workout supplements or whatever. Those are all naturally occurring in the human body as well, in all sexes and across all genders. It's known that there are certain man made contaminants in the environment that can affect the levels of these androgens and therefore cause biological responses, either by interacting directly with the androgen receptor, which then kind of bypasses all the androgens and just acts as another androgen also interacting with the androgen receptor and causing that effect, or by changing the levels of the androgens in that pre-regulator stage. So PFOA, which is a type of PFAS, has long been speculated to have some role in this androgen dysregulation. We don't know whether it was at the androgen receptor level or at the level before that, at the pre-receptor level. So we do know that PFOA was able to directly interact with certain other receptors in context of lipid biogenesis. And it's been heavily associated with non-alcoholic fatty liver disease. However, we have seen epidemiologically, so like at just people level, there does seem to be an association between PFOA and androgen dysregulation. However, we don't really know where it's occurring. So what we set out to find was if it was occurring at the androgen receptor level or at the pre-receptor level. And what we found was that it actually was occurring at the pre-receptor level. It was able to interact with aldehydo reductase 1C2, which is one of those pre-receptor androgens. And what that does is it takes a potent androgen, an androgen that would normally interact with the androgen receptor to elicit that biological response, and inactivates it. So that enzyme is responsible for the decrease of that potent androgen. However, PFOA was able to inhibit it and therefore not allow it to do its duty of decreasing the levels, therefore keeping the levels high. And that's how it was interacting with the receptor.
I believe you utilized HPLC for analyzing the androgens. Could you talk about why that was the right method of analysis for you?
I talk about toxicology, I talk about biochemistry, and then also the techniques that we use in the lab is another like area of expertise kind of. And we do use HPLC a lot. We use a lot of different types of chromatography, and we also use mass spec. We tend to think of ourselves as a little bit more of an analytical lab. In this particular case, why it was correct to use chromatography, and specifically the type of chromatography we used, is that there are assays to see how androgens are being metabolized. However, there was no particular assay to see how our androgen was being metabolized by this enzyme in the reaction that we wanted it to occur in. So we had to develop an assay from scratch, kind of, with a lot of, background already having been done. And to do so, I guess like we had to use HPLC. Now, what was really interesting about this is that oftentimes for androgens, you can detect them using UV or VIS spectroscopy. However, that was not possible for the androgen that we were particularly interested in, which is DHT. It has absolutely no chromophore, so we weren't able to detect it in any way. Therefore, we had to label it somehow. And what we decided to do was radioactivity, since we do have a radioactive detector in our lab. And it was really interesting to do that because while radioactivity has been like used with HPLC in the past, it isn't really used that commonly, particularly in our field. It's not used very commonly. So it was really fun, honestly. It was like kind of a challenge to see how to do like both sides of it.
I can say that when we were exchanging messages, I
Mhm.
believe you used the term radiochromatography, which is something I had never come across before.
Yeah, the technique that we decided to use is radiochromatography, which is using radioactivity to detect the analyte after passing it through the chromatography column, right? So you use the chromatography to still separate the analytes, but then you use radioactivity to actually detect them. So therefore, your analytes need to be radioactive. That was a lot of fun, honestly. A lot of new techniques that we had to use. A little like new and old because I feel that a lot of the work kind of is moving away from radioactivity. So it's like rediscovering old. One of the first challenges with using this technique was that we needed to get standards for the analytes, and they don't sell a lot of the things that we wanted. um, so the main androgen we are able to get commercially in radioactive form. However, the metabolites that we were then looking at downstream are not sold commercially, so we had to synthesize those. And thankfully, since we work on this very specific pathway with these enzymes, someone maybe like 10 years ago in the same lab as me actually found a way to synthesize it with almost 100% yield using one of our enzymes that we have in the lab, and he mutated that enzyme so it can have 100% yield. He did it in a slightly different context, but looking at that, I was like, oh, we can use that to synthesize our standard that we need. So we did that. And then obviously we looked at it through HPLC to make sure that it was pure. And once we had that, we had now both standards. Then comes always the challenge of chromatography, which is being able to separate it. And if you look at androgens in general, they're very similar. They're like the difference between some of them are literally stereochemistry of a hydrogen if it's like in the up position or in the down position. So they're very similar. So separating it was probably the hardest part, I would say. And that's true of most chromatography, but definitely multiple months were sunk into that, just separating it using different columns. We did finally achieve it with a C8 column, playing around with the flow, different solvents and everything. And then radiochromatography has a additional layer of the detector. So the detector uses liquid counting or scintillation counting, which is a way essentially of taking the radioactive atom and converting the radioactive energy from that atom that it emits into light energy using the solvent that it's mixed with. So you need to pass fresh scintillation solvent at the same time that you're passing the chromatographic solvent. So you have two different flows that you're kind of playing around with. If it's too slow on the scintillation side, then you're not going to have clean peaks, because even if your analytes are being separated correctly, you're going to have a delay or like some kind of carry over from the previous analyte in the detector. So you have to play around with both of them. Also, if you do it too fast, there's never really like a problem technically of doing it too fast, but you're going to use a lot more of the scintillation fluid and it's pretty expensive. So you want to play with both of those things. But finally, we were able to get a method that worked on all sides, and it was really satisfying when that is able to happen. The most important thing about the PFAS work is that we were looking at a particular reaction, and we're looking at the impacts of the PFAS chemical on that like paradigm that I was explaining with the androgen receptor. The current work that I'm doing is following up on that original assay I developed with radiochromatography and expanding it to more androgens and looking at metabolism downstream of those androgens, how those cells metabolize different androgens, and then we were detecting them and analyzing, quantifying those androgens.
That's great to hear. Could you talk about your sample preparation in detail, what the workflow looked like?
For the sample preparation, we get our samples from cells. So after treating the cells and letting them do what they do to the androgens, we take a sample from the media of the cells. And then we extract out the androgens. And we do this with a fairly simple extraction with diethyl ether. We shake them and then we extract out just the organic layer into a separate tube, and we dry that down. We do that multiple times. And then after we dried all of it down, we reconstitute it with 60% acetonitrile, which is just compatible with the chromatographic method that we use. So pretty simple sample preparation on that end.
When you say dried down, is that using nitrogen blowdown?
Yeah. Yeah. So we've experimented with a couple of different types of drying down, and yeah, we do find that nitrogen is probably the best.
Very good. Could you talk about what your starting volume is, and then are you going all the way to dryness, or how does that look?
Yeah, so extractions use 2 milliliters of ethyl acetate. So when we isolate those 2 milliliters, we then dry completely down to zero. And since we do that multiple times, usually 2 or 3 times, you're looking at 4 to 6 milliliters drying down completely. And thankfully, diethyl ether is a very like volatile solvent, so it dries down pretty quickly. Another reason that we use radioactivity is because it has minimal sample like manipulation. So another way that we can also detect androgens and that we do in the lab is using mass spectrometry as the detector essentially. However, androgens don't ionize particularly well, so you have to derivatize them first. And that derivatization takes probably like a day, but it does add like certain steps into the manipulation of the sample. However, if you start out with a radioactive androgen from the start, you have to do pretty little to it because it's already labeled essentially. So, the general workflow of this experiment, I would grow up cells for the experiment. I would add radioactive androgen to them. I would then take different time points to see how they're doing, what they're doing to the androgen at different times. So you can kind of follow it across a timeline.
What challenges did you face developing and implementing the HPLC workflow in general?
Yeah, so a little bit like how I mentioned, I think the biggest challenge is the fact that androgens are very similar to each other. They're all derived from cholesterol. All the steroidal hormones are derived from cholesterol, and then further you can break those down. And androgens are very, very similar. And since we're looking at a particular androgen and its metabolites, each of those metabolites only change it so slightly. Oftentimes, it's just the removal of a double bond, or the androgen that the reaction we're looking at actually is taking something from a ketone to an alcohol. So that's the only difference. And while you might think, okay, well, if it's changing it from a ketone to an alcohol, then it's going to change the polarity. You're correct in thinking that. However, it doesn't work exactly how you always think. For some reason, some of our alcohols would come out before the ketones. So it's like you never really can expect it until you do it. And that's really the thing that has been that that's really what I've learned the most in doing this type of work is that you can try to predict how everything's going to go. Like, oh, if you raise the flow rate or if you change this concentration of solvent, this is like theoretically what should happen. And then you actually do it and you're like, oh, never mind. That for some reason it didn't happen. And then you'll think of like a third or fourth reason as to why it happened that way. It's never what you think it's going to happen. But I particularly really love chromatography separations for that reason because it is very much like trial and error when you're developing that method, which I just think is probably aggravating at the time, but also really fun looking back on it. As I mentioned, also the analytes are very similar, and we have two analytes that are only stereo. The only difference between them is a stereochemistry difference in their hydrogen. So one of them has a hydrogen in like the upward position to the plane that you look at, and one has it to the bottom position of the plane you look at. So you would think that those slight differences you couldn't even tell when you run it through a chromatographic method, but you can. And interestingly, those were not the difficult ones to separate for some reason. The difficult ones to separate were actually like two separate ones that should not have looked alike at all.
What else would you like people to know about your work with androgens?
That's a good question. I think the main take home message for anyone who's not in science, or maybe it's just a science enthusiast, but maybe not actually a scientist, is that everyone has androgens, even if we normally think about them in a male context or a workout context. We all have androgens, so this work impacts all of us. Another thing to really think about is how the environment really does impact those androgen levels and how that does lead to a lot of unknown effects. Some of the bigger picture that is the reason that we do this work is that it affects diseases such as polycystic ovarian syndrome, in which patients have higher levels of androgens, and that is thought to maybe affect some of their outcomes. We also know that higher levels of potent androgens lead to things like prostate cancer. So, across everyone, all humans, androgens play an important role, and if they're dysregulated, they have a potential to cause a lot of harm.
That makes sense. Have you ever done PFAS work that was connected to any of the EPA methods?
No, not that I'm
Okay.
aware of now.
Yeah, it's just PFAS is such a broad,
Yeah,
broad topic.
yeah, it definitely is. I feel like every time I go to like any conference, people know immediately what that is, and people are like way more keen to talk about that.
Which conferences do you typically attend?
Society of Toxicology and American Chemical Society under the Chemical Toxicology division.
And that's probably where we met, was at an ACS.
Probably. Yeah, we also have methods to actually detect PFOA and PFOS.
Hmm.
And you would think that I use those all the time, but my work is more like on the biological side. So I'm really detecting androgens. But I always think it's interesting when I like talk to other people because that's often what they think about what that I do. Like, and we do have those methods. So sometimes when they ask me, when people are like asking for collaborations or something, I'm like, oh yeah, we do like we can do that. I particularly don't do that, but we do have scientists who do that.
Well, I suppose, and I'm not a chemist, but just to speculate, I would suppose it's the drinking water testing from an environmental standpoint that gets a lot of the media attention,
Yeah,
where actually figuring out what the PFAS is doing and the impact on our bodies is like a whole separate
yeah,
study that's just as critical, right? But it's also totally different.
yeah. It's like different. It's related. I'm always really like happy to see any sort of PFAS work done. It is very like interesting because I don't know. It is just different, but it is like also similar enough that I always feel like happy when I see it.
Yeah, it's an important area. Andrea, I really appreciate the time today. Thank you so much.
Yeah, thank you.